And one more thing, Afro Humanist, just to show you the difference between your education and mine. You believe in the theory of evolution, right? Can you tell me how you get new anatomical features? That would be anatomy if you don't know that big word. Okay. Can you tell me how you get new anatomical features from random mutations, ma'am? And that's just one of a whole bunch of questions that not even your scientists can answer. But you believe evolution is true. Can you tell me how you get life from non-life, ma'am?
Sorry, it was what is a mutation? What does it do?
How do you get new anatomical features?
How do you get new stuff? So the way you get new stuff is really cool, and this gets into a field called EVO-DEVO, evolutionary developmental biology. And the really cool thing is that there isn't like, it's a common misconception that like there's a gene to make eyes, there's a gene to make legs, there's a gene to do X, there's a gene to do Y. And what we actually see is that there are genes that kind of set patterns. So like there's a gene that says extend away from the midline of the body. There's a gene that'll say fold inwards. And the kind of the timing and the location of the expression of all these different genes in combinations gets you the different structures. And so, for example, I'll use the eye as an example, because this is the nice thing about eyes is that you can look at all the different organisms that have the ability to like detect light in some capacity, and you could see basically the evolutionary history of eyes across those different organisms. So there's a group of proteins. It doesn't matter how they work. They're called opsins. They detect light. That's all that matters. And you can express those genes. They're evolved from another set of proteins. It doesn't matter what the other set does. It doesn't matter. And basically, you could picture a flat surface, right? And you detect light. Okay, like you could detect light, dark. That's fine. But what if you had, instead of a flat surface, what if you had the signal that says fold inwards? Well, folding inwards is a very common signal developmentally because that's a way to increase surface area. And increasing surface area is like, we like that. That's great for absorption or exchange. So like gills, intestines, you want to fold in. So let's say you fold in on the surface. Now you have your opsins in, a cup. Well, now that's useful because now you can detect the direction of the light, right? Is it coming from this direction or is it coming from this direction? That's useful. Now, along with those opsins and the folding pattern, you can have a layer on top. It's easy to have two layers of cells, and for one to say fold in and the other one to not fold in. Well, now you can have a translucent layer on top and a folded in layer on the bottom. This is starting to sound like an eye. And you can go through these different stages without needing any specific like new genes, but you're just changing the regulatory pattern of how they're expressed and where and when during embryonic development, and you get a much more complex structure. And you can look at this from top to bottom. So you could look, for example, at the development of limbs as the same kind of thing, where you're looking at like extend from the body. Okay, well, the simplest things that extend from the body have like little nubs, right? Think about like a caterpillar leg. It doesn't have like true legs. It's just like little nubs, right? But then if you can look at other groups, arthropods, mature arthropods, insects, spiders, that kind of thing, well, they have true legs. Mammals, true legs. Reptiles, true legs. Well, the really cool thing is they have the same genes that say build a leg here, but it's not a gene that says build a leg here. All that gene says is extend here. And if you express that gene for a long enough time, it causes the tissue to split and grow in two directions. So that's how you get like digits, for example, in your fingers. So our arms aren't just one continuous thing. It's at some point it splits into multiple things that grow away from the body. So and I apologize, this is not a short answer, but the short-ish answer to how do you get new information is you have a pre-existing thing, and we could talk about where that comes from because there have been lots of experiments on that, but you have a pre-existing thing that does a thing, extend from the body, build X, build Y, and you change the pattern of where and when that's expressed within a multicellular organism, and that results in a new feature in that organism.
And for how long, right? It's where, when, and how long is it expressing.
But it's